Hello students, today we will learn about cryopreservation, germplasm conservation and cryopreservation. This is the second video of this germplasm conservation and cryopreservation. Let's start. Here we learn about addition of cryoprotectants and pre-treatment, how we can do pre-treatment. Cryoprotectants are the compounds that can prevent the damage caused by the cells by freezing or thawing. The freezing point and supercooling point of water are reduced by the presence of cryoprotectants. As a result, the ice crystal formation is retarded during the process of cryopreservation. There are several cryoprotectants which include dimethyl sulfoxide, that is DMSO, glycerol, ethylene, propylene, sucrose, mannose, glucose, proline, and acetamide. Among these, DMSO, that means dimethyl sulfoxide, sucrose and glycerol are most widely used. Generally, a mixture of cryoprotectants inside of a single one is used for more effective cryopreservation without damage of cells or tissues. Now, let's know about freezing. The sensitivity of the cells to low temperature is variable and largely depends on the plant species. Four different Four different types of freezing methods are used. First is slow freezing method. The tissue or the requisite plant material is slowly frozen at a slow cooling rate of 0.5 to 5 degrees centigrade per minute from 0 degree centigrade to minus 100 degree centigrade. 0 degree centigrade to 100 degree centigrade, and then transfer to liquid nitrogen. The advantage of slow freezing method is that some amount of water flows from the cells to the outside. This promotes extracellular cell formation rather than intracellular freezing. As a result of this, the plant cells are partially dehydrated and survive better. The slow freezing procedure is successfully used for the cryopreservation of suspension cultures. Rapid freezing method. This technique is quite simple and involves plugging of the vial containing plant material into liquid nitrogen. During rapid freezing, a decrease in temperature of 300 degrees centigrade to 1000 degrees centigrade per minute occurs. The freezing process, the decrease in temperature should be minus 300 degree to minus 1000 degrees centigrade per minute occurs. The freezing process is carried out so quickly that small ice crystals are formed within the cells. Further, the growth of intracellular ice crystals is also minimal. Rapid Rapid freezing technique is used for the cryopreservation of shoot tips and somatic embryos. Now, third freezing method is stepwise freezing method. This is a combination of slow and rapid freezing process, and is carried out in a stepwise manner. The plant material is first cooled to an intermediate temperature and maintained therefore about 30 minutes, and then rapidly cooled by plugging it into liquid nitrogen. Stepwise freezing method has been successfully used for cryopreservation of suspension culture, shoot apexes and buds. Dry freezing method. Fourth point is dry freezing method. Some workers have reported that the non-germinated dry seeds can survive freezing at very low temperature in contrast to water imbibing seeds, which are susceptible to cryogenic injuries. In a similar fashion, dehydrated cells are found to have a better survival rate after cryopreservation. Now, let's know about the storage. Maintenance of frozen cultures at the specific temperature is as important as freezing. In general, the frozen cells or tissues are kept for storage at temperature in the range of minus 70 degree to minus 196 degrees centigrade. However, with temperature above 130 degrees centigrade, ice crystal growth may occur inside the cells, which reduces viability of cells. Storage is ideally done in liquid nitrogen refrigerator at 150 degrees centigrade in the vapor phase or at minus 190 degrees 96 degrees centigrade in the liquid phase. The ultimate objective of storage is to stop all the cellular metabolic activities and maintain their viability. For long-term storage, temperature at minus 196 degrees centigrade in liquid nitrogen is ideal. A regular and constant supply of liquid nitrogen to the liquid non-nitrogen refrigerator is essential. It is necessary to check the viability of the germplasm periodically in some samples. Proper documentation of the germplasm storage has to be done. The documented information must be comprehensive with the following particulars. First is taxonomic classification of material, history of culture, morphological potential, genetic manipulations done, somaclonal variations, culture medium, growth kinetics. Now let's know about thawing. What do you mean by thawing? Thawing is usually carried out by plugging the frozen sample in ampoules into a warm water. That is, temperature should be 37 degree to 45 degree centigrade. Bath with vigorous swirling. By this approach, rapid thawing, that means at the rate of 500 to 700 degrees centigrade per minute, occurs, and this protects the cells from the damaging effects ice crystal formation. As the thawing occurs, the ampoules are quickly transferred to a water bath at temperature 20 to 25 degrees centigrade. This transfer is necessary since the cells get damaged if left for long in warm water bath. For the cryopreserved material where the water content has been reduced to an optimal level before freezing, the process of thawing becomes less critical. Now let's know about the reculture. In general, thawing germplasm is washed several times to remove cryoprotectants. This material is then recultured in a fresh medium following standard procedures. Some workers prefer to directly culture the thawing material without washing. This is because certain vital substances released from the cells during freezing are believed to promote in vitro cultures. Now, let's know about the measurement of survival or viability. The viability or survival of the frozen cells can be measured at any stage of cryopreservation or after thawing or reculture. The technique employed to determine viability of cryopreserved cells are same as used for the culture. Staining techniques using triphenyl tetrazolium chloride, Evans blue, and fluorescence diacetate are commonly used. The best indicator to measure the viability of cryopreserved cells is their entry into cell division and regrowth in culture. This can be evaluated by the equation number of cells by organs growing whole divided by number of cells divided by organs thawed into 100. Now let's know about plant regeneration. The ultimate purpose of cryopreservation of germplasm is to regenerate the desired plant, appropriate plant growth and regeneration. The cryopreserved cells or tissues have to be carefully nursed and grown. Addition of certain growth promoting substances besides maintaining maintenance of appropriate environmental conditions is often necessary for successful plant regeneration. Now let's know about the cold storage. Cold storage basically involves germplasm conservation at a low and non-freezing temperature of 1 to 9 degrees centigrade. The growth of the plant material is slowed down in cold storage in contrast to complete stoppage in cryopreservation. Hence, cold storage is regarded as a slow growth germplasm conservation method. The major advantage of this approach is that the plant material is not subjected to cryogenic injuries. Long term cold storage is simple, cost effective, and yields germplasm with good survival rate. Many in vitro developed shoots or plants of fruit tree species have been successfully stored by this approach. Example: grape plants, strawberry plants, virus-free strawberry plants could be preserved at 10 degrees centigrade for about 6 years with the addition of a few drops of medium periodically once in 2 to 3 months. Several grape plants have been stored for over 15 years by cold storage by transferring them yearly to a fresh medium. Low pressure and low oxygen storage. Let's know about how we can preserve the seed in low pressure and low oxygen storage. As alternative to cryopreservation and cold storage, low pressure storage LPS and low oxygen storage LOS have been developed for germplasm conservation. Now, let's know about first LPS, low pressure storage. In low pressure storage, the atmospheric pressure surrounding the plant material is reduced. This results in a particular decrease of the pressure exerted by the gases around the germplasm. The lower partial pressure reduces the in vitro growth of plants. Low pressure storage systems are useful for short term and long term storage of plant materials. The short term storage is particularly useful to increase the shelf life of plant materials. Example: fruits, vegetables, cut flowers, plant cuttings. The germplasm grown in cultures can be stored for long term under low pressure. Besides germplasm preservation, LPS reduces the activity of pathogenic organisms and inhibits spore germination in the plant cell system. Now let's know about low oxygen storage. In the low oxygen storage, the oxygen concentration is reduced, but the atmospheric pressure, that should be 260 mmHg, is maintained by the addition of inert gases, particularly nitrogen. The partial pressure of oxygen below 50 mmHg reduces plant tissue growth, that is organized or unorganized tissue. This is due to the fact that with reduced availability of oxygen, the production of carbon dioxide is low. As a consequence, the photosynthetic activity is reduced, thereby inhibiting the plant tissue growth and dimension. Now, limitations of low oxygen storage. The long-term conservation of plant materials by low oxygen storage is likely to inhibit the plant growth after certain dimensions. Now let's know about applications of germplasm storage. The germplasm storage has become a boon to plant breeders and biotechnologists. Maintenance of stock cultures. Plant materials of several species can be cryopreserved and maintained for several years and used as and when needed. This is in contrast to an in vitro cell line maintenance, which has to be subcultured and transferred periodically to extend viability. Thus, germplasm storage is an ideal method to avoid subculturing and maintain cells or tissues in a viable state for many years. Cryopreservation is an ideal method for long-term conservation of cell culture which produce secondary metabolites like medicines. Disease-free plants or pathogen-free plant materials can be frozen and propagated whenever required. Recalcitrant seeds can be maintained for long period. Conservation of somaclonal and gametoclonal variations in cultures. Plant materials from endangered species can be conserved. Conservation of pollen for enhancing longevity. Rare germplasms developed through somatic hybridization and other genetic manipulations can be stored. Cryopreservation is a good method for the selection of cold-resistant mutant cell lines which could develop into frost-resistant plants. Establishment of germplasm banks for exchange of information at the international level. Thank you. Finished. Any questions regarding germplasm? I will clear. Please kindly write in the comment box and subscribe my channel to know more videos of biotechnological topics. Thank you.